So I would like to thank ARM for inviting us today and giving me the opportunity to share what we are doing at LogicBio. I need to remind everybody that this presentation will contain some forward-looking statements, so please refer to this slide. So LogicBio is a clinical stage biotechnology company focusing on delivering treatment for early onset genetic diseases based on two platforms. One gene editing platform, one gene delivery platform. The company has been started in 2016, and in 4 years we've been happy to get our first IND for a gene editing platform. We raised a total of $180 million, mainly through equity and some upfront from collaborations. And as we are developing our pipeline, we are also being able to create some partnership with 3 biopharma companies, based on our gene editing and gene delivery platform. So our first gene editing platform is called GeneRide, and it's a promoterless nuclease-free gene editing technology which is designed to provide a very targeted and precise insertion of a missing gene in the genome. We have the second technology platform called SAVI. It's a next generation synthetic AAV platform where obviously we search for, we engineer capsids for better tissue tropism, high yield, and we also select them in clinical relevant models. So our most advanced program is in the clinical stage, as I said before. It's called Sunrise, and it's an in vivo knock-in for the treatment of MMA. We've been announcing the first patient in June 2021. We've been showing some early data in October of last year, where basically we've been able to claim the first ever gene editing in a pediatric patient, and the first ever knock-in, which means the insertion of the gene in a specific locus of the patient. Unfortunately, in February of this year, we had to announce that the FDA put the program on clinical hold due to 2 adverse events called thrombotic angiopathy, TMA. And we are not disclosing, any of the discussion, but we are working very actively with the FDA and our DSMB to solve this problem, and we will share more when appropriate. And as I said, we have a pipeline that we are developing by ourselves or with some partners that I'm going to describe now. So we believe GeneRide is well suited to intervene in very early, in young patients, and so in early onset diseases where it's important to intervene before some non-reversible sequelae, some tissue damage, non-reversible damage occur and can lead to long-term complications. We've initially decided to address diseases where we can target the liver, as we realize that there are very few programs in gene therapy and gene editing targeting liver diseases in pediatric patients. So first program in MMA, as I said, and recently in December 2021, we announced our second development candidate for Thyrocynemia type 1, and I will show you some data in a minute. We have also some proof of concept in other diseases such as Crigler-Najjar and Wilson disease, alpha-1 antitrypsin, and a few others, and we continue to develop new proof of concept based on GeneRide. And so some internally and some with our partner Daiichi Sankyo that we are not disclosing to date. But with the gene delivery platform, we've been able to out-license one of our capsids, and we collaborate with a pharmaceutical company called Cambridge Pharmaceutical on the gene therapy program for Fabry and Pompe disease, and they have an option for 2 other indications. So GeneRide, in a nutshell, it's, as I said, promoterless nuclease-free. It's composed of 3 main elements: the transgene to express the gene of interest, It's coupled with a small peptide of 21 amino acids called 2A peptide, and it's flanked by very long homology arms. These homology arms are driving the integration in a specific locus of the genome, close to or right behind a highly expressed gene. When we target the liver, we go after the albumin. As you know, it's the most expressed gene in the liver and maybe in the body. So by homologous recombination, we insert at the very end in a non-disruptive manner at the end of the albumin locus. And after the transcription, what the 2A allows us to do is a bicistronic or bicistronic protein expression, which means with one read of one mRNA, we can express two proteins. Obviously the protein of interest, here in orange, but also in the case of the liver, when we target albumin, we express one albumin, which is tagged with a small peptide, with the 2A. And that's something we can measure in the blood. So it's a very easy way to confirm that you have integration exactly where you thought you would have integration. So great technology-related biomarker, which provide us to do some more invasive seq. So there is another interesting phenomenon we discovered with GeneRide in some more than one disease, actually, and I'm going to talk about two today. First, in MMA, it's called Selective Advantage. What it is, after a limited number of cells being modified after the injection, the modified and healthy hepatocytes are taking over more and more of the liver and replacing the sick hepatocytes. As you can see on the picture on the left, and if you start with the quadrant on the top left, there is in few brown dots, that's the number of cells modified after the injection. But as the time passes, you have more and more hepatocytes being modified. And on the right-hand side, you can see in orange the albumin 2A concentration increasing in the blood, and it mirrors the number of hepatocytes we are growing in the liver. We saw the same thing in Thyrocynemia type 1, but with one major difference, it happened much faster. You may have noticed that in the previous slide, it took about 4 to 5 months in mice to plateau, around 40 to 50% of the cells being modified in the liver. What we see here on the left in Thyrocynemia is after a few weeks, 4 weeks, depending on the dose, we can repopulate 100% of the liver. And that's a pretty interesting data, because Thyrocynemia type 1 is a true hepatocyte disease. And what we do very quickly, we do what we call a molecular liver transplantation. There is a treatment for this disease. It's called NTBC. The treatment carries some side effect and is at risk of very low compliance. But what you can see here, we can restore the liver function very quickly after injecting the mice very early on without the need of NTBC. And we arrive to levels which are even better than the standard of care to date. And same for succinate acetone, which is one of the toxic metabolites. We can dramatically reduce the number of, the levels of succinate acetone after only one injection in the early days of the mice. And all these things correlate with preclinical biomarkers such as life expectancy, weight, and obviously, the succinate levels. So I'm going to close the page on GeneRide, and maybe to focus on the second part, which is our gene delivery platform called SAVI. And here are 3 main components. The first one is bioengineering of synthetic capsids. And the second one is about how to select this capsid. As you know, most of the capsid has been selected in black 6 mice, in normal mice, and they have been selected for their propensity to target a specific tissue in the mice. Now, years later, we know that some very good capsid to target, for instance, the liver of a mice, are not so good or not so efficient, when we take them to NHP, and even more so when we take them to humans. So the idea here was to select some capsid in more clinically relevant model. For instance, in the liver, we use a mice with which has been humanized with a human liver. or 90+% of the liver has been repopulated by human hepatocytes. And we select the capsids for their ability to target the human hepatocytes and not the murine hepatocytes. And the last piece is process development. As you know, it's one of the Achilles tendon in gene therapy, even if I think we have done a lot of progress in the past years. But it's important to, make progress here, and we have developed at LogicBio some proprietary technologies where we have totally reinvented the way we produce AAVs, and which give us very, very good data. So, talking about the yield, here you can see, the significant improved yield in comparison with published results with multiple platforms. Basically, we gain about 1 log when we compare other platforms to our new system. Not only, it's good for yield, but it's also very good for the purity of the product. And we have also developed some downstream processes which help us to have very good ratio in between full, partial, and empty, because we look at these 3 types of categories. And we have obviously very, a large panel of very elaborated assays to accurately target this, measure, all these elements. And the last thing I wanted to say, is that we've also noticed that with our new process development, we not only have a more pure product, but we have a more potent product. And we have some hypotheses to explain why. So maybe to conclude, as I said, we have 2 platforms, a gene editing and a gene therapy platform. The gene editing one is called GeneRide, and we have, to my knowledge, the only one today doing knock-in in the clinic with an in vivo delivery. As you know, other gene editing technology are doing mostly knockout with 1 vector. To do knock-in, they would need 2 vectors. Here, we use only 1 vector. So the program is on its way, and hopefully we'll be able to restart the Sunrise program as soon as possible. And on this gene delivery platform, which gives us a very interesting capsid that we are already taking, in project with partners and also internally. We have, I think, developed a very interesting way of producing this capsid, which gives us, I think, a very interesting competitive advantage and will allow us to produce at a very limited cost. And nothing would have been possible without the team. And so, as my accent would not help you to predict, I'm based in the US, in Lexington, Mass. Thank you very much.